Thank you very much for the kind introduction, Imogen. So I'm a medical oncologist. I specialize in the treatment of melanoma, but I'm also a Wellcome Trust funded scientist. So I run a research lab studying how the immune system defends us against cancers, including melanoma. So these are my disclosures. And I'm conscious that this is the first session of the day after the Congress Party. And usually this is a sort of very coveted session when we go to academic conferences. And so I'll sort of try and ease us into clinical trials. Now, when I'm in the clinic and I mention clinical trials to my patients, I get some very positive reactions and I get some sort of slightly skeptical reactions like, whoa, doctor, I don't want to be a guinea pig. And I think that's a very reasonable sentiment. So if you look at the types of clinical trials that we have, we've got these phase 1 trials. And in a phase 1 trial, this is often the first time that a new drug, has been tested in humans. And it's absolutely right that, you might have some trepidation. So the purpose of a phase 1 trial is really to test whether drugs are safe or not. By the time a trial or a drug gets to phase 2, we know that they're relatively safe. And what we're trying to test then is whether or not a drug actually works. And then if you pass that bar, you then get on to what we call these phase 3 trials. So these are trials where we know the drug works, we don't quite know how well it works, and we want to test whether or not it works better than the gold standard that we have today. Now, by and large, the trials that you'll encounter in melanoma clinics up and down the country are going to be phase 3 trials. So these are trials where we're fairly sure the drug is relatively safe, we're fairly sure it has some effect, and we just want to know, is this better than the gold standard? And this is why trials are really, really important. Without trials, we would never get new drugs licensed. Then I guess, why are trials important for you as a patient? Well, they give you access to potentially better drugs. And sometimes they may give you access to drugs where you might not have any. And of course, you may help others in the future. But I think a really important point, which doesn't always come across, is that when you're on a trial and the drug that's being used is paid for by the pharma company, in my view at least, this means you're saving your NHS-funded line of treatment. And that's really important because the NHS only funds certain lines of treatment. And if you can save that, that's a benefit in itself. So that was the gentle primer. Now we're going to get really stuck into the data. So I mentioned that phase 3 trials test drugs that we as doctors and pharma companies, etc., think are better than the current standard. Now, to really show this, a phase 3 trial has to have a lot of patients, usually several hundred. And that means it costs hundreds of millions of pounds. So that also tells you we've got a pretty good feeling that these drugs are going to be better. But that doesn't always pan out, okay? So you can sometimes get drugs that lead to worse outcomes. That happens occasionally, but there are data monitoring committees in these trials which will stop the trial early to try and reduce the amount of harm that might come to patients. More often, you get trials that show that the drug is no better than what the current standard is. And in fact, you can see here in melanoma, this was the case for many, many decades. So this is sort of how we present trial data. It's called a survival curve, and I'm sure you'll see many more of these, so I'll take some time to go through this. So on the x-axis here, the horizontal axis is plotted time. That's the time from when you receive treatment. And on the y-axis, the vertical axis, is how many patients are alive. So for a very good drug, what you want to see is that line staying right up at the top. So patients staying alive as time goes on. If you have a bad drug, that line, unfortunately, will go down as patients die. When you have two drugs and you're comparing them, what you're looking at is, what do those lines look like next to each other? And if they're overlapping like they are here, it means one drug is no better or no worse than the other. Okay? So that's really important to bear in mind. It's only through these clinical trials, though, that we've managed to find some of the revolutionary treatments that we have today, like the immunotherapies. And this began with this first drug here, anti-CTLA-4, also known as ipilimumab. So I think some of you will have had this, or some of you will have heard of this. And you can see here, there were 2 drug treatments here that contained ipilimumab, and it's very clear. There's clear water between those lines and this line down there, which did not include ipilimumab. So ipilimumab was the gold standard drug for a very short period of time for treatment of advanced melanoma. It was soon after overtaken by the anti-PD-1 class of drugs, and these are drugs like nivolumab and pembrolizumab, which many of you will have heard of before. And you can see here, there's clear water between anti-PD-1 drugs and ipilimumab. More recently, we've had other combinations that have given us slightly better benefits over anti-PD-1. And here you can see the newer combination, anti-PD-1, anti-LAG3, so nivolumab and relatlimab, that gives you a slight improvement in certain situations. Now, what I want to focus on in all of these graphs is this right-hand bit of the graph. And this is what we as oncologists refer to as the tail in the curve. And this is when these survival curves begin to level off, and they begin to look almost horizontal. And what that tells us is, if you make it to that tail in the curve, after usually about 2 to 3 years, patients stay alive forever. And that really says to us, you're forever until the end of this graph. And that says to us that, you know, most of these patients are probably cured. So this tail in the curve is really, really important. And this has really only been achieved in cancer with immunotherapy. So with therapies that modify our body's immune system to reject cancer. And that's why, for the rest of this talk, I'm going to focus on immunotherapy trials, and that's also why I spend a lot of my time in the lab trying to study this. So, it's long been known that the immune system controls melanoma, and this is what melanoma looks like under the microscope. And I'm no expert at microscopy, unlike Dr. Matthew in the back there, who's going to give you a masterclass in about 20 minutes. So instead, I'm going to explain this in really quite simple terms. In the maroon here are immune cells. And in fact, they're mostly a type of immune cell known as a T cell. And you can see they're surrounding and containing these melanoma cells in the middle. I'm going to zoom in now to the front line here. I'm just going to expand that a little bit more and show you what's happening between this immune cell and the cancer cell. So there are many types of immune cells. Most of them that I'm going to be talking about are these T cells, and in particular, I'll talk about one type here, just for simplicity, called the killer T cell. So these cells do exactly what they say on the tin, which is they kill cells which they don't like, which they perceive as a threat, and they do so by recognizing these red flag molecules that these cancer cells will express. Most of the time, this is happening inside our bodies. So these T cells are pruning back tumor cells on a very microscopic level to protect us from full-blown cancers. And in fact, some of you may have noticed moles which come and then go away forever. And that's actually your immune cells pruning back a mole that's on the turn. And of course, the problem is, unfortunately, life is a bit of a game of chance, and there's always a chance that cancer cells will somehow escape immune killing. And this is one of the key ways in which they do so. They express molecules on their surface which tell that immune cell, hey, stop, don't kill me. And this means that cancer cell escapes this killer T cell surveillance. And of course, chances build up with age, which is why most cancers are a disease associated with advancing age. Now, as Ross said last night, knowledge is power. And the knowledge of how these signals work mean that we can begin to make drugs that disrupt these signals. And that's exactly what drugs like ipilimumab, nivolumab, pembrolizumab, etc. do. They block this stop signal from the cancer cell to the killer T cell. And that lets the killer T cell do what it needs to do. And in particular, it's these anti-PD-1 drugs, pembrolizumab and nivolumab, that are incredibly effective at doing this. When I say incredibly effective, I mean they work about 40% of the time. Okay? Of course, that means that more than half the amount of time, these drugs don't work. And one of the reasons why they don't work is that red flag signal is lost. Okay, so these cancer cells begin to evolve, and they lose that red flag, so they're no longer visible to the immune system. And this resistance, particularly to the anti-PD-1 drugs, which I'm going to refer to as anti-PD-1 resistance, is the biggest challenge in the treatment of melanoma, particularly for advanced and metastatic disease. So I'm going to focus on some of the trials that really target this. Now, this is one way in which cancer cells can evade the drugs that we currently use, but it's not the only way. And if I were to zoom out a little bit, you'll appreciate how many different ways cancers have of evading the drugs that we use. So we've zoomed out a bit, and we're just going to look at what the tumor microenvironment looks like. So this is the killer T cell and the cancer cell. There are some good cells in this picture. So you've got these other immune cells here called dendritic cells, or DCs, and their aim is to support the role of the killer T cell, to make it a better killer. But unfortunately, you've got a lot of bad actors. So you've got these myeloid-derived suppressor cells, or MDSCs. You've got cancer stem cells, and these guys will directly support the growth of the cancer. And on the other side, you've got these bad immune cells here, B-regs and T-regs. And what they do is they inhibit, or they stop the killer T cell from killing. Of course, the net effect there is that it promotes tumor growth. Then you've got these cancer-associated fibroblasts. They prevent any more of these T cells from getting into the tumor. So you can see it gets pretty complicated, pretty fast. And you could look at this and say, oh, how are we ever going to overcome this? So I actually take the optimistic view and say, well, look, there's a lot of processes here that we can target with drugs. And remember, almost all of our current drugs target this one process. And by doing this, we've unlocked a 40% cure rate. So if we can hit all of these other processes, imagine where we can get to. So with that in mind, I want to focus on 3 trials which I think have the promise of tackling this critical issue of anti-PD-1 resistance. And that's these 3 trials here. So there are many ways of tackling anti-PD-1 resistance. One way of doing this is through what I like to call this intellectual takedown. And here, what we're doing is we are rationally rewiring some of these cellular and immune processes to boost how these T cells can kill cancer. And the first 2 of the trials that I'm going to talk about are examples of this. So the first one is the Telluride 006 trial. So this trial focuses on the good guys, and it says, can we do anything to boost the action of these good guys, these dendritic cells, to try and help them help killer T cells kill? And the trial, or the trial drug, is called ICON 1001. And what it does is it flips a switch on these dendritic cells and turns them on and superactivates them. And this switch is flipped on by these molecules called TLR7 and 8. And what it does is it actually mimics a viral infection. So the dendritic cell thinks there's a virus that's infected it, and what it does is it turns itself on and it supercharges that T cell. And that T cell then becomes a much more efficient killer. And that's the theory. Does it work? So let's look at the trial evidence. Here's one of the phase 1 trials that have looked at ICON 1001 in combination with pembrolizumab. And this is what we call a waterfall plot. And again, you'll probably see more of these today, so I'll take some time to go through it. Each bar here is a patient, and if the bar goes down, it means the tumor in the patient has shrunk after treatment. And if the bar goes up, it means the tumor has grown. And we set this relatively arbitrary cutoff of 30% reduction. So if your tumor reduces by 30%, we say the drug has worked. It's caused a response. So you can see here already that some patients have responded by this 30% cutoff. And importantly, this patient here in the dark green is a patient with melanoma who had previously had anti-PD-1 therapy, pembrolizumab, and had progressed. So this is the type of patients that we're very, very worried about, because we have no good treatments for patients who progress on anti-PD-1 therapy. And you can see this patient with the ICON drug has managed to respond, and in fact the tumor reduced by about 50%. And if you look at all of the phase 1 trials that have been done with this drug, about 20% of patients with melanoma who had anti-PD-1 resistant disease had a response, and that's pretty good. Okay? It's not spectacular, but it's pretty good. What about the side effects? So this is a table of the side effects, and you can see most patients on this drug got minor side effects, these grade 1 or grade 2 side effects. So these are side effects which you notice, which may interfere a little bit with your day-to-day activities, but aren't really something we're very worried about. These grade 3 side effects are the ones that you get a phone call for, where your oncologist might say, well, you might need to come in, you might need to slow down your treatment a bit, etc. So these are the ones we tend to worry about, and you can see with the ICON drug, it's relatively safe with that respect. Only a few percentage of patients getting these moderate to severe side effects. So on the basis of this relatively reasonable tolerability and the evidence of efficacy in this anti-PD-1 resistant melanomas, we're now running this phase 2-3 trial at Guy's and at other centers in the UK. And in this trial, patients with advanced untreated melanoma get randomized to either pembrolizumab alone, which can be considered as one of the standards of therapy, ICON 1001 at a medium dose or a high dose in conjunction with pembrolizumab. And this is a 1-to-1-to-1 randomization. So you have a 2-to-1 chance of getting the trial drug. And really importantly, the pembrolizumab in this trial is funded by ICON, which means that you as a patient will preserve your NHS-funded line of treatment going into this trial, regardless of which arm you're randomized to. Right. So next up is the IGNITE-3 trial. So this is a really cool trial. So if I just come back to the tumor microenvironment here, what the IGNITE-3 trial does is it focuses on these two processes. And it uses a drug which is actually called an oncolytic virus. And that's just a very fancy way of saying a virus which kills cancer cells. And this is the virus. Some of you may recognize it. Anyone? So this is the most successful virus in the history of human civilization, and it's the herpes virus, okay? And everyone in this room will have had some form of herpes virus. So I see some surprised looks here, but it is, everyone will have had one form of herpes virus. So the most common form, of course, is the cold sore, okay? So herpes causes cold sores, and if any of you have had a cold sore, you'll know the sort of natural history of it. You get a little bit stressed or run down, you get a cold sore, but then by itself, after a few days, it goes away. And then it comes back sometimes. But what that tells you is that the base virus, this virus here, the herpes virus, is actually really, really safe. We live with the herpes virus throughout our lives. It comes and it goes, and it doesn't normally cause any problems. And this is the life cycle of herpes. So, these are the cells on your lip, for example, and that's the virus there. And when you're not stressed, your immune system is working, and it's making these antibodies, and it's protecting you from herpes infection. But when you get stressed, these antibodies go away, your immune system gets suppressed, and the herpes virus comes out, and it's able to infect these cells in your lip, for example. And after an initial tussle with some of the intracellular defenses, represented by this protein PKR there, what herpes does is it wins out, and it begins to turn that cell into a virus production factory. And eventually, new viruses burst out from that cell, and that cell dies. And these new viruses come out into the space in between these cells, and they look for more cells to infect. Now, that sounds pretty dramatic, sounds a bit dangerous, but it's not really, because this process doesn't go on forever. If it did, our entire face would become a cold sore. So what eventually happens is that these other healthy cells here bolster their internal defenses so that herpes can't get in. Then you become a bit less stressed, you recover, your immune system comes back, these antibodies here come back, and they actually wipe out that herpes virus. So that's pretty cool, right? But what does herpes have to do with cancer? Well, by understanding exactly how herpes works and lives, we can actually begin to engineer what it does, and we can make it do really cool things, including redirecting this potent capacity to kill cells to a cancer cell. So that's the native herpes virus. And what scientists have done is they've changed it a bit. And they've made this drug called T-VEC. And some of you may have heard of T-VEC, some of you may well have received T-VEC. And what T-VEC is it's a herpes virus that has a deletion in a gene that makes it overcome PKR. In other words, this T-VEC virus can no longer overcome normal cell defenses. And this is really important because by the time a cell becomes a cancer cell, it loses those defenses. It no longer has PKR in it. And what scientists have also done is they've inserted an extra gene into this T-VEC virus to help stimulate the immune system. And I'll come on to that. So you've got this herpes virus here. Because of this gene deletion, it can no longer infect normal healthy cells, and that makes it really, really safe. So when you get treated with T-VEC, you don't get cold sores because your healthy cells will stop that virus dead. But these cancer cells are highly susceptible to T-VEC, okay, because they don't have that defense protein PKR. And when T-VEC goes in, the other thing it does is it begins to make a lot of chemicals that stimulate your immune system. That red gene there that's been engineered actually encodes for proteins that stimulate the immune system. And it does it in two ways. It can stimulate the immune system by secreting chemicals. It can also stimulate the immune system by increasing this expression of that red flag molecule there, making these cancer cells very, very visible. And of course, it's a herpes virus, so it kills. It kills the cells that it infects. It infects those cancer cells really efficiently. The T-VEC is a great drug, but it's licensed. It's not investigational. We use it at Guy's very extensively, but it's the next generation of T-VEC that I'm really excited about. And what scientists have done is they've taken T-VEC, it's a virus that works, so why break it? And they've added to it this nuclear warhead right at the top. So that red molecule makes T-VEC a super killer now. And what it does is it gives T-VEC this ability, so now it's called RP1, by the way, and it gives this RP1 virus this ability to break down the walls in between cancer cells. So what happens is you just have to infect one cell with this virus, and it will spread or propagate itself across all of the neighboring cancer cells. And you can see that makes it a super efficient killer. And of course, this also lets the virus hide from these antibodies. Normally, for that virus to infect another cell, it's going to have to come out, and that will reveal itself, and the immune system will get rid of it. Without having to come out the cell to spread, the virus becomes super, super efficient at spreading within the tumor. And of course, it still does everything that T-VEC can, so it can stimulate the immune system by making these chemicals here, and it can make these cancer cells express these red flags. And of course, it can kill these cancer cells. So, in theory, a very good killer. And this is the big picture. RP1 stimulates these dendritic cells here with the chemicals that it makes. That supercharges the killer T cell. The RP1 also increases expression of that red flag molecule, making cancer cells much more visible to these supercharged killer cells. And, to top it off, it can directly kill these cancer cells and probably these cancer stem cells too. So, in theory, a very, very good killer. Does it work in real life? So this is a phase 2 trial now of RP1 in patients with advanced melanoma who have failed anti-PD-1 therapy. So remember, this is a population of patients where we really have very poor treatments. And there were 140 patients in this trial. And the trial is set up like this. So patients had to have tumors that were injectable, at least one, okay? And you can see there's a tumor in the groin here that was injected with RP1, and patients had up to 8 injections. But there were also tumors that were not injectable. So if you have a very deep tumor in your liver or in your abdomen that you can't easily reach with a needle, you didn't have an injection there. But the responses were measured both in the injected tumor and in the non-injected tumor. And that's really important because the only way that you could have a response in a non-injected tumor was because the virus stimulated your immune system, and your immune system went to attack that tumor. And that's important because we know that when you activate the immune system, you can get cures. Here's the waterfall plot. So there's about 140 patients here, And you can see most patients actually responded to treatment. And if you use the arbitrary cutoff of a response by 30%, about 1/3 of patients had responses to RP1. And this is really important for two reasons. One is these patients were resistant to anti-PD-1 therapy. And our current standard of care drugs for this population of patients gives responses in about 10 to 20% of patients treated. So here we're looking at about 30 to 35%. Secondly, you can see that patients, even those with advanced disease, so M1b, M1c, and M1d means patients with lung metastases, liver metastases, or brain metastases, they all responded. And that's a major improvement over the current generation of viruses that we have, like TVEC. Now here's a separate waterfall plot, and this is a really important one. So this is in all the patients who had a response, they measured the degree of that response, both in the tumor that was injected, which is in blue, and the tumor that wasn't injected, which is in red. So you can see nearly every patient had responses in their tumors that were not injected. And in fact, if you look at this patient here, he had one tumor, he or she had one tumor that was injected and 8 that were not injected, and all of them completely disappeared. So if you get to the bottom, it means a 100% shrinkage. So this is a very potent drug. What about the side effects? Well, it's actually pretty good. So if you look at these grade 3 side effects, these moderate to severe side effects that we get worried about, the rate of grade 3 side effects is actually not much more than the virus plus nivolumab alone. So you're not adding very much toxicity to the nivolumab that you're using with this drug. So highly safe. Okay. Ooh. So on the basis of that, both the tolerability and the efficacy, particularly in these anti-PD-1 resistant melanomas, we're about to open this phase 3 trial of RP1 at Guy's and other centers in the UK. And in this trial, patients get randomized to RP1 plus nivolumab or physician's choice therapy. And what this reflects is the fact that actually there is no standard therapy for patients who have progressed past anti-PD-1 therapy. So we all think there are certain drugs that may be a bit better or a bit worse, but there's no accepted standard. So the drug company will fund all of these, and importantly, they will fund drugs that are not funded on the NHS. And these are good drugs. So it means that if you go on this trial, even if you don't go on the trial arm with the investigational drug, you're very likely to get slightly better drugs than you would do on the NHS. So it's a win-win. So I've talked a lot about intelligently designed drugs that rewire the immune system to try and fight cancer. But there's more than one way to skin a cat. And another strategy that's proven reasonably effective is this American military doctrine of overwhelming force. And you're gonna hear a good example of this later on today from Dr. Furness, who's gonna talk to you about TILs. But rather than stealing his thunder, the last trial therapy I'm gonna introduce is actually a combination of intelligent design and overwhelming force. And that's the SUPREME trial here. And Susanna and Melanoma Focus have been really instrumental at pushing this trial forward in the UK. So the SUPREME study is very elegant because it just focuses on this core business here of a T cell killing a cancer cell. And in this study, patients with anti-PD-1 resistant melanomas have blood taken, and they have these killer T cells taken out from their blood. Now, the problem with these killer T cells, of course, from the blood is not many of them will recognize melanoma. So the company behind this trial, Immatics, has taken a T cell with this special receptor on it that does recognize melanoma. And in fact, it recognizes melanoma by the expression of this melanoma-specific protein called PRAME. And what they've done is they've taken this receptor and they've actually grafted it on to these T cells they've taken from your blood. And that redirects all of these T cells from the blood to see melanoma cells and to hunt them down and hopefully to kill them. And importantly, and unlike TILs, these T cells that we're getting from your blood are healthy, fresh T cells. They're not exhausted, they're ready to kill. They haven't been sat in a tumor for years and years. So what then happens, and this is where the overwhelming force comes in, is these T cells, or these reprogrammed T cells, now known as IMA203, are put back into patients. And they're put back in at a dose of about 1 to 10 billion cells, okay? And that's a huge number of T cells. The total number of T cells normally in anyone's body is about, well, in their blood, is about 10 billion. So we're putting back as many T cells back into a patient's blood. Does this work, though? Well, here's the data, and it's pretty exciting. This was a phase 1 trial published last year of 33 patients with anti-PD-1 resistant melanoma. So these were patients whose melanomas grew despite being on anti-PD-1. And here's the waterfall plot. 2/3 of patients had a response to this cellular therapy. Now, I just want to set this in context. Our current gold standard in the UK would give you a response rate of about 10 to 20%. And even with TILs, which aren't funded or licensed in the UK, but which you can get in the US, you'd be looking at 30 to 40%. And in this trial, we're looking at about 60%. Now, granted, this is very small numbers, okay? So you can't take too much away from this. But what I would say is, included in these patients here are patients with melanomas that are extremely hard to treat, uveal melanomas, where essentially you do not see responses with any drugs that we have on the market. Mucosal melanomas, again, very difficult to treat melanomas. It seems to work for all of these patients. And of course, there's no such thing as a free lunch, and that's doubly true in the world of medicine. And in terms of side effects, it's absolutely true that this drug is a tough drug, okay? It's much more harsh than ICON 1001 or RP1. And in particular, in red there are these side effects that we worried about, these grade 3 or 4 side effects. But what I would say is they're mostly associated with this lymphodepleting chemotherapy. So this is a chemotherapy that we have to give with this drug. Because remember I said I'm putting in 10 billion T cells. Well, you have to make room for those T cells. And the way that's done is you get chemotherapy to actually reduce some of your own T cells to make a little bit of room. And that chemotherapy is quite toxic. So the majority of these side effects are related to that chemotherapy. The good news there is those chemotherapy side effects tend to be very temporary. It takes you a few weeks to get over them, and then you're okay. So on the basis of this, we are hoping to open this trial across the country in the next few months. And this is a phase 3 trial of patients with PD-1 resistant, anti-PD-1 resistant melanoma. And again, it's a 1-to-1 randomization. So you either get the IMA203 or you get a physician's choice standard arm. And again, the drug company will fund drugs which are not funded in the NHS. And these are very good drugs in the second line. And really surprisingly, in the US, not here because it's not licensed, they will fund treatment of TILs in the comparator arm. So that's incredible because it gives you a very active drug in the comparator arm, a very expensive drug in the comparator arm, and actually that tells me the drug company is pretty confident that it can beat TILs, which are currently, globally at least, the gold standard for second-line therapy. And really importantly, actually, and this is what I really like about this trial, it's very geared up for patients. So in the process of screening for the trial, if it happens that you develop a brain metastasis, usually in a trial, and anyone who's been on a trial will probably attest to this, usually in trials, if you screen with a brain metastasis, you are removed from that trial because we don't know how these drugs work in the brain. The drug company has actually made a separate trial and said, if during the course of screening you happen to develop a brain metastasis, you will just get the trial drug, which I think is a really, really patient-friendly design. How much time have we got? Have we got another 2 minutes? 1 minute. Perfect. So a lot of potential great treatments on the horizon for anti-PD-1 resistant melanoma. We're getting more and more good drugs to choose from. And the question is, how do we use them? So in the last few minutes, or last 1 minute, I'm going to consider this. So broadly speaking, there are 2 schools of thought on how to use cancer drugs. You can either throw the kitchen sink at it, or you can keep your powder dry, all right? So one argument for throwing the kitchen sink is this belief that when you combine drugs together, they help each other out, or they synergize, so that the combination of two drugs is better than using them separate. And this has been a very long-held belief, and there's certainly some science behind this. But unfortunately, the clinical data does not support this. And what we actually see is that when you use two drugs, if you use them in sequence or combination, the chance of response is the same. In fact, it's just additive. And in fact, actually in real life, it's a little bit less than additive. Nonetheless, I think it's true to say that if you use the combination, you do get better responses up front, okay? And of course, by proxy, you might think also that it leads to better survival. So why not just do this? Well, because combinations give you toxicity, and Dr. Board will tell you a little bit about that later on today. The converse, of course, is, well, why don't we just use the drugs sequentially? If you use drug A and it works, you're spared the toxicity. It's fair to say that there is a higher chance of it not working compared to combination. But you can always then just use drug B at that point. And actually, you're probably likely to get a similar cure rate. The worry, of course, is that for you as an individual patient, that you might not get a chance to use drug B because your cancer has progressed a lot quicker than you thought it would, or that your doctors thought it would. The reality, though, is that this probably doesn't happen as often as we think, because there is no evidence yet to conclusively say that one option is better than the other. So for me, I think it's really important, as we get more and more drugs, we begin to consider how we use these drugs in combination or in sequence. And those trials really need to be designed to sort of answer this question. So I think that's where I'd like to end. And finally, just give a plug to the Melanoma Focus Clinical Trials Finder. I think this is a really incredible resource for patients and for healthcare professionals alike. Thank you.